All right, we're going to talk about the cerebellum today. This should definitely be important to you all, since it's so important for movement. Coordinating our movement, keeping us balanced as we carry out movements. Because when you pick up things, you're gonna put weight, and you might fall forward if you're holding onto something while bent over, let's say. So you're going to anticipate that, and you're going to shift weight back. Of course, you're not going to do that. Your cerebellum's going to do that without you having to think about it. So this is how we go from uncoordinated toddlers to little kids who can run around on the playground and swing and slide and all that jazz. And you don't have to think about it. And that's because there's several different outputs and inputs to the cerebellum. So it has a really good idea of what's going on, probably before the cortex does, in fact. And it has an idea of what the cortex is planning. It can send feedback to our motor cortices to refine their pattern so that we choose the right upper motor neurons. It can provide output to the brainstem nuclei, so the descending non-conscious outputs, the vestibulospinal tract, reticulospinal tract, so we can stay balanced and carry out these routine bilateral movements, respectively. The way that it does that is by distributing the workload in different functional regions, and that's what the first part of the lecture is about. There's three broad zones in the cerebellum that help us keep our balance, carry out anticipatory movements, and then refine our movements so that we don't make the same mistake in the future. How these all work is going to be the topic of the second part here, where we go through the different cell types in the cerebellum and how they can modify their synapses whenever some error occurs. So we got really three different areas of the cerebellum that are going to get different types of input and have different types of output to affect the motor cortex as well as the brainstem, and ultimately then control the output of the spinal cord so that we have coordinated movements. That's the whole purpose of the cerebellum. So a mid-sagittal view here, just so we get some idea of what our cerebellum looks like. The cortex, so the topic of the second part here, the circuitry of the cerebellum is really just in these little gray areas here. And you can see it's highly convoluted, just like our cerebral cortex. And that's just to increase the surface area, because we're packing a lot of neurons into here. About 80% of all of our neurons live in the cerebellum. Somewhere around 70 billion or so, just in these little gray areas, these little folds we call folia there. So there's where the magic of motor coordination happens. You'll notice these white matter tracts on the inside. This is what's going to connect the rest of our nervous system to the cerebellar cortex, and the cortex down to the deep nuclei. We're looking at the central portion here, so this will be the vermis, and we have these larger lobes, a little more lateral to that. There's a primary fissure here that's going to separate the anterior and posterior lobes. We'll have a look at those in the practicum and see the vascular supply for those regions. But we got three basic divisions that I think you should know. And they give you some idea of what they're going to communicate with based on their name. So the vestibulocerebellum is all about the vestibular system. There's direct input from vestibular afferents. There's also input from the vestibular nuclei. And the output targets, for the most part, vestibular nuclei. There's also the reticular nuclei as well. This is going to be from the inferior portion of the vermis, and then this hidden part that we're not seeing yet, I'll point out in just a moment, the floccular nodular lobe. If we were to take off this cover here, there's an ancient strip of tissue, the floccular nodular lobe, that's hidden down in there. Its deep location should suggest to you that it's going to have direct output to the targets. So it acts as its own deep nucleus. The inferior vermis is still going to hit a deep nucleus. We'll get to that in just a bit. So the vestibulocerebellum, back and forth, reciprocal connections with the vestibular system. The spinocerebellum is all about coordinating our motor output. So it's going to have input from those spinal cerebellar tracts that we went over in lecture 9, I believe. So review those. These are going to relay our non-conscious proprioception, also any of the spinal reflexes going on, so the movements that we're carrying out and our body position. So the spinal cerebellum has a great idea of what's our body position. It's going to comprise the vermis here, right in the center, so this is going to be involved in controlling our trunk. Like the inferior vermis, this more superior portion here, same output. It's going to target the same deep nucleus and then eventually target the reticulospinal and vestibulospinal tracts. There's also some ascending input from the spinal cerebellum, and that's going to be for our paravermal hemisphere. So this strip right here, just next to the vermis. So more medial portions than this cerebellum. Since they're in different parts of the cerebellum, they're going to have different targets. You might guess that the target here would be the cerebrum, and you'd be correct. So the cortex is going to be the major input and output here for our cerebrocerebellum. We'll have input from the cortical pontocerebellar tracts, so from the cortex to the pons to the cerebellum. In other words, planned movement. The output here is largely going to be to the thalamus and then to the cortex as a result. So here this is more about refining movements, motor learning. We'll see that. We're going to pull the inferior olive in with this tract as well. So we'll see some pictures where it'll make a little more sense. The input and output is going to be by white matter tracts called the cerebellar peduncles. So if we were to turn that cerebellum around and cut it off from the rest of the brainstem, it would look something like this. So now we're looking, here we're looking from the back. Now we're going to turn around and we're looking from the front. So the brainstem should be sitting here. We have a nice pons here, and the pons are going to provide major input through the middle cerebellar peduncles there, the red ones. The superior cerebellar peduncles are for the most part output, ascending output. So if we're targeting the thalamus or the red nucleus or the tectum, we're going through the superior cerebellar peduncles. So we're going upwards. That name should make sense. Superior. Inferior would be the downward projections to the brainstem. Middle is very simple. That's your input from the pons. This is major input too. The tracts from your cerebrum to the pons is about 40 times larger than from your cerebrum down to the spinal cord. So in the cerebellum, we have most of the neurons in our brain. This is where also most of the motor output goes. About 20 million axons in that cerebropontine tract. And the cerebrospinal tract, one of the three major tracts you need to know as a clinician, only about 500,000 fibers. So far less than we have here. And that's because the cerebellum is so important for coordinating movement. We have to dedicate a lot of neurons to that so we can refine our movements and we don't have to worry about the details up here. We just think, move my hand forward. This takes care of the rest. Do keep in mind that the input can also come from the inferior and superior cerebellar peduncles. There's that double cross for our anterior and rostral spinal cerebellar tracts. That comes in the superior. And the inferior is going to provide mostly the input from our vestibular nuclei or the primary vestibular afferents and also any of the lower brainstem nuclei. The other spinal cerebellar tracts come in the inferior cerebellar peduncle as well. So here's our roads in and out. Superior and inferior would be the output upward and downward, and the middle is very straightforward. Just that major input, so that we have an idea of what actions we're planning. Based on their input, they can create different body maps. So different parts of the cerebellum are going to have unique body maps, and there's at least a few of these. The color is showing you which part of the body it kind of pertains to. You're going to see head, arm, leg, in that order. Kind of how it's arranged in the body. Head, arm, leg. Different areas are going to be able to handle different types of information and deal with different functions. For example, keeping our balance. Well, that's going to be more in the central regions here because they're going to communicate with the vestibular and reticulospinal tracts. So that we have this automatic change in our center of gravity as we go to pick something up or we change body position. And the activity of these neurons doesn't have to affect other neurons that are involved with, let's say, coordinating a complex task, like running up a scale. If you're going to do that, you don't have to think about your posture. You also don't have to think about the automatic finger movements because it's all built in right here. But by having distributed body maps, we can carry out all the major functions of the cerebellum at the same time. We can keep our balance. We can carry out non-conscious anticipatory movements, and we can have our conscious movements be carried out in a smooth fashion. We can do all those things at once and more because we have distributed body maps. They don't have to interfere with one another. If we only had one map here, we could only do one thing at a time. That wouldn't work out very well. Maybe this will be helpful to you for your studies. Here I've organized the three parts of the cerebellum and provided you with just kind of a very basic summary of where they are in the cerebellum, the major input to them, the deep nuclei that they target. So these nuclei are going to have different targets here, which are listed down below, and that's going to tell you about its function. For example, your vestibulocerebellum. Here we should be thinking vestibular system, balance. We're probably not going back to the cortex other than to let it know that everything is fine. The major output here is really going to be those that are involved with maintaining posture and also that vestibulo-occiput reflex. Notice vestibulo is right in there. This should make sense because of the targets. We'll get into these later when we talk about eye control, but there are nuclei within our brainstem that control where we gaze. We call them gaze centers. There's 4 of them to control left, right, up, and down. These are targeted by the vestibulocerebellum so that whenever we have movement of our head, we can keep our eyes fixed. It also allows for proper smooth pursuit so that your eyes can move smoothly rather than how they normally move, which is jerky, generating saccades. So when you go to look from one side of the room to the other, it's instantaneous. There's not a smooth movement. Try to move your eyes smoothly without tracking an object. You can't do it. It's impossible. These targets should make sense because of what we're targeting up here. We're targeting the vestibular nuclei. We're going to find out that these are connected to the gaze centers. That should make sense. They're also going to generate those vestibulospinal tracts. The vestigial nuclei, well, these are going to target the reticular formation, help recruit reticulospinal tracts here. So it's all about keeping us balanced. And we can do that because we have a good idea of what's our head position. What's the movement of our head right now? Are we tilted a little this way? That's going to matter. The spinocerebellum. A little more complicated because you'll see there's a bunch of different inputs here. There's one descending input, so we have some idea of our planned action. That's the cortico-pontocerebellar tracts. Everything else is going to be descending. So what's going on in our spinal cord? What's going on in our head? We're going to relay that information into the vermis and that paravermal area, so the more medial portions of the cerebellum. The output there is going to be from the vermis, the vestigial nuclei, and then from the paravermal area, the interposed nuclei, globose and emboliform. Those are going to target different areas. So the vestigial nuclei still target our reticulo and vestibulospinal tracts. So we get these automatic movements to adjust our posture and to carry out some well-defined action like walking. Remember the walking that we saw for cats that had their cerebrum removed? So they're able to still use these tracts and these tracts. So that reticulospinal tract is going to get recruited by our spinal cerebellum based on what's going on in the rest of our body and head. What are we planning on doing? That's what's going to determine how we carry out our steps. And we're going to make sure we stay balanced as well. Now the interposed nuclei are pretty similar to the dentate. We'll see there's a little subtle difference that kind of makes a difference there. But we're going upward here. So here we're going out those superior cerebellar peduncles to higher regions, the ventrolateral thalamus. This is very important for coordinating motor output. So whenever we're deciding which motor neurons to recruit, that depends on which thalamic neurons we're exciting, because those are the neurons that are going to send excitatory feedback to recruit the correct motor neurons. The red nucleus will provide another descending track down to our upper limbs. So we can control automatic movements of our upper limbs, our lower limbs, and then head, trunk, lower limbs for balance. This portion is all about anticipating changes in body weight as we carry out movements. So these automatic movements that get carried out before you do the thing you're really thinking about. That's what we're going to carry out here. So before we eventually hit back on the cortex via the thalamus, we've already recruited these other descending tracks before then, so they can anticipate what we're going to do and make sure we don't fall over. There's also some activation of antagonistic muscles so that we can terminate movements in a smooth fashion rather than jerky. So you'll see tremor, intention tremor, whenever you have damage to your spinal cerebellum, because you can't properly recruit, essentially, the brake. You're not activating the antagonistic muscle to smoothly terminate a movement, and so instead it's an abrupt stop, and then the elastic properties of the muscle kind of stimulate stretch reflexes, and you get kind of a little back and forth jerk, because muscles are elastic. And so if you don't properly terminate their movement by recruiting that antagonistic muscle, they have a tremor back and forth. The final portion would be the cerebellum. Most lateral portions, so we're out there in the lateral hemispheres. The input is fairly straightforward here. It's planned action. So from the cortex to the pons, pons to the cerebellum. So the cortical, ponto, cerebellar tracts. Exactly what it sounds like. Cortex, pons, cerebellum. The target here is going to be the dentate nuclei. Very similar output. We're still hitting the ventrolateral thalamus, just like over here. We're still hitting the red nucleus, a slightly different portion of it. And we're also going to get the superior colliculus here so that we can move our eyes around. The big role here is in planning movement, because the major output is going to be ascending to the thalamus to help control the activity of the motor cortex. So this is going to help us carry out movements in a smooth fashion, because we'll select the correct upper motor neurons. It's also very important for learning, motor learning that is, potentially other kinds too. While the cerebellum is largely involved with just coordinating movement, it seems to have more complex functions than that, particularly with language. We won't go into that, but you will see some language deficits that go beyond just simply the inability to coordinate your mouth and tongue and larynx. There's some word associations that get lost. So there's a little more to the cerebellum than movement, but let's not worry about that. Let's just worry about movement, because that's more than enough. And that's the major function. Refining movements. Let's see these in a picture form, and then we'll have our three divisions, I think. So here we're just looking at the output from that vestibulocerebellum. There's direct output to the vestibular nuclei. That's what this little cartoon neuron is showing us. Now that inferior vermis is also going to target our vestigial nuclei. So here's our 1, 2, 3, 4 deep nuclei. We always group our emboliform and globulus nuclei and call them the interposed nuclei. So it's 3, but it's 4. Isn't that fun? The vestigial nuclei are going to target our vestibular nuclei as well. So a couple different ways of hitting the vestibular nuclei so we maintain balance. We're also going to recruit our reticular formation so we can activate the reticulospinal tract and carry out those well-patterned bilateral movements. So maintain balance and carry out that same thing that you do every day, day in, day out. There's some ascending output as well so that the cortex knows that everything's handled, but this is minor. This is what our vestibulocerebellum is all about. Recruit these lowest structures down here so that we get that non-conscious motor output. The spinal cerebellum is still going to involve our vestigial nucleus here, so that's still playing a role. We're also going to bring in the interposed nuclei from that paravermal region. So a little lateral to the center, we're going to target the interposed nuclei. So we still have our descending output, so we get our our automatic, non-conscious, anticipatory movements. But here we can also start to target the upper limbs. So we'll hit the red nucleus. And there we have our output down to the spinal cord, so the root row spinal tract to control some upper limbs to some degree. And then ascending input to the thalamus. The thalamus is going to then communicate back and forth with the motor cortex. These are reciprocal connections here. So the cortex has a plan, runs it by the thalamus. Based on input from the cerebellum, that thalamus is going to approve certain parts of the plan, maybe not others, and send a more refined version on up for the final output via the cortical spinal tract. So both upper ascending and descending pathways here for our spinocerebellum. For this, just see the last slide. The cerebrocerebellum is going to use the dentate nucleus. This thing is actually massive. This is the biggest deep nucleus here. This is all about ascending input. There's one descending tract from our red nucleus. So here we're targeting a slightly different cell population. Rather than projecting downward to control upper limbs, we project downward to the inferior olive. The inferior olive is then going to provide those climbing fibers so that we rearrange the synapses out here in our cortex. These are active if we make an error, if we fall down, for example. Part of what's going to help recruit the inferior olive would be ascending input along the anterolateral pathway. So remember that there's a bunch of offshoots in our anterolateral pathway. One of them is going to be the inferior olive. So if you're trying to walk, you fall down, it kind of hurts. You're going to sense that as an error, in part because of output from the dentate nucleus to the red nucleus down to the inferior olive. You'll also sense that more directly because you'll activate nociceptive afferents in the anterolateral pathway. So as we're going on up to form our spinocerebellar tract, we'll provide a little offshoot to say that hurt. And because that hurt, you probably shouldn't carry out that particular pattern of movements again. You should change it. I don't know how to change it, but whatever you did, stop doing that. We'll see how we do that in the second part of this talk. But the reason that the cerebrocerebellum is so important for motor learning is because of this, the ability to recruit the inferior olive, which provides very strong input to our Purkinje cells, and that causes them to weaken those active synapses that created that error. You fell down, don't do it again. But for the most part, cerebrocerebellum is going to be about the cerebrum. So we're going to hit the ventrolateral thalamus again because this is how we get to and from our motor cortex. So there's still that back and forth. Just like in our spinal cerebellum, the cerebrocerebellum is going to refine the pattern of activity in the motor cortex by targeting the thalamus. And with all of these acting together, what we're going to do is carry out some automatic movements so we maintain balance, and we're going to coordinate our conscious movements so that they're not jerky and we don't have any sort of tremor. So our motor cortex is going to convey planned actions. Here's what I think I should do. How should I do this best? That's what we're going to figure out here. Because the cerebellum has far more neurons than our entire cortex, let alone just the motor cortex. A whole lot more processing power down here. So we go from the cortex on down to the pons via the middle cerebellopeduncle into the cerebellum. Couple types of outputs. Really, there's two major functions of the cerebellum. We're going to produce some anticipatory movements. So we have an idea of what's going on in our body because we have input from the spinal cord, for example. Down here we know what's going on in our upper limbs and our lower limbs. We have an idea of our sense of balance right here. That, along with our planned actions, goes in. We're going to target different areas, like we just went over, in order to refine our movement. That's going to be mostly in that more lateral portion. Is the dentate, goes on up to the cortex via the thalamus. We're going to get some automatic movements and we'll maintain balance. So here's an example of an anticipatory movement that will help you maintain balance. Before lifting heavy weight here, what this guy's going to do is, of course, activated biceps so that they contract so you can lift heavy weight. When doing that, you're going to be putting heavy weight on the front. You need to bear weight so that you don't fall over. And this, which of course he's not thinking about, is activated first. Because as we send that plan down to the cerebellum, there's that immediate output from our spinal cerebellum that knows, I'm going to lift heavy weight. Make sure I don't fall over. So we activate those descending non-conscious tracts. Vestibulospinal tracts, the lateral vestibulospinal tract, for example, is going to activate our lower limbs and make sure we don't fall over. We don't have to give the moment's thought. We just think, I want to lift heavy weight. When we do that, we make sure that that added weight forward doesn't make our whole body move forward. Have you ever tried to pick something up and you thought it was way heavier than it was? Just, whoa, my goodness. Or you're pulling on something and suddenly it becomes unstuck. And you can see where when you're not anticipating that change in balance, movement's kind of tough. You might fall over. Maybe you thought there was going to be a stair there and there wasn't, or you thought there wasn't a stair and there was. Those anticipatory movements that didn't happen, you find out how useful they are in instances like that. And that's what our cerebellum is going to handle. It knows we're walking downstairs, there should be one here, so we have a different set of anticipatory movements than we would if we were just walking on flat ground. And you don't have to think about it, because we automatically recruit those non-conscious descending pathways. Now we also send feedback so that we can refine our movements, so that ascending output that hits the thalamus or the red nucleus and then inferior olive, that's going to make sure that whenever we do carry out a movement that involves multiple joints, we can do it in a smooth fashion. Are you guys impressed? This is pretty good, right? And I don't even have to think about it. It's a wonder I couldn't give more dates. I would always do that. And that's actually very tough to do without a cerebellum, without a functioning cerebellum. Even just this is tough. So I think this will be a little more clear whenever we see some damage. So whenever there's damage to the more medial portions of the cerebellum, well, that affects the more medial portions of our body. Remember, we're there in the vermis, the flocculonodular lobe, those parts that are dealing with balance. So you might expect that you're going to see balance issues. So when they walk, well, that automatic coordination of the lower limbs is gone. Their sense of balance is essentially gone, non-conscious sense of balance. And so they look like they're drunk. And people with cerebellar damage will sometimes lose their jobs. You'll hear some stories like this because their superiors thought they were coming into work drunk, even though they weren't, because they walk like this. Also, there's a little bit of speech slurring because the cerebellum helps coordinate speech. So it's unfortunate. So they have what we call a drunken gait here, so they try to keep their feet wide so they can maintain balance, but everything is uncoordinated. You can see it right there. And their ability just to stand still with their eyes closed is lost. Are you guys impressed now? I can do that for at least a minute. And that's what you do in the Romberg test. And when you look at the sway that someone has when they have damage to the cerebellum compared to a healthy subject, they're all over the place. So if they stand on a force plate and you can sense their center of gravity, with cerebellar damage, they're going to be moving all over because they don't have that automatic coordination of their lower limbs to maintain balance. So now they're always having to think about it and consciously try to maintain balance. And it's a lot more difficult when your cortex is trying to carry out the movement. If you've ever learned how to do anything, any sort of movement, like throw a dart, shoot a basketball, do a backflip, whatever, the first time you do it, it's very difficult. But as you practice and practice and practice, and you start to then recruit your cerebellum, now there's no thought to it. And you carry it out much better than you did when you were thinking about it really hard. Like if you're learning how to serve in tennis or something like that. It's terrible the first 1,000 times you do it, because you're thinking about it. Movements are much better when you don't think about them, and you let the much more powerful cerebellum handle it. So there's a couple tests you can do here. You can have them walk one foot in front of the other to exaggerate that drunken gait, because now they can't have a wide-based gait to maintain balance, so you can more easily see when they start to sweat. More lateral portions of the cerebellum, well, we're going to see different deficits. More lateral portions are going to control more lateral portions of the body, So we should expect our limbs to be affected more so here. So the appendages. So here's a fella. He's trying to get a date. You can see. All right, great on the right side. Now the left side. So he has left cerebellar damage. Again, ipsilateral. We should remember this from the last unit. You notice that intention tremor. So it's difficult for him to control his agonist, antagonist motor neurons so that he doesn't sway at a joint. So it's hard for him to hold it still. And so he gets this kind of back and forth tremor there. And you can see his movements very uncoordinated. It's hard for him to coordinate each different joint into one smooth motion. Every joint is essentially its own thing he has to think about. And it's very tough to think about all those joints at once. So there's a few different tests you can do here. Basically, you have them move either their upper or lower limbs and have a look. Is it coordinated? Do they have any sort of tremor? There'll be deficits in the timing, the amplitude, so it won't be a nice consistent back and forth like this. And that's because they don't have that non-conscious coordination going on via the cerebellum. There are other deficits that you'll see. Eye movements will be uncoordinated because of the intimate relationship between our vestibular nuclei and our gaze centers, and of course the cerebellum and our vestibular nuclei. When there's cerebellar damage, there's issues with eye control. So they'll have these involuntary movements. This is gaze-evoked nystagmus. So when you're looking extremely left or right or up or down, it's hard to hold that extreme position. You see these improper corrective saccades there. And then we'll see some smooth pursuit in a little bit, I do believe. Okay, so you'll notice that smooth pursuit is not just a nice smooth movement of the eyes. There's these saccades. There's these very fast involuntary movements of the eye. And that's because of the inability to properly regulate the vestibulo-ocular reflex, which is really important for smooth pursuit. Now, of course, the inability to have a good sense of balance and control your sense of balance, well, that's going to lead to a little nausea. There can be difficulties coordinating speech, especially more lateral portions. The big takeaway is that there's really no change in strength, no change in reflexes, no change in how they perceive their body. So conscious perception is all there, but that non-conscious, automatic coordination is lost. And so you'll see uncoordinated movements, but the patient won't feel any different other than, well, maybe a little nausea here. But their two-point discrimination, for example, or pinprick, all that's going to be preserved. You'll see no difference there. Depending on where the lesions are, you'll see a change in balance or a change in the ability to move their limbs. Strength will be preserved, though. Conscious proprioception preserved. Any questions? All right, cool. Then let's go through these and then we'll talk about the synaptic circuitry. Well, then let's talk about the circuitry of the cerebellum and how input becomes output and how we modify output when something goes wrong. That's probably the most important part here. There's kind of two important elements. One would be the filtering that occurs in the cerebellum. So, canceling out weak activity so that we get a greater signal to noise. We want black and white. We don't want shades of gray so much. We want strongly activated neurons to be active and weakly activated neurons to be silenced. So we get a clear pattern. That's our first major idea. And we're going to see a couple steps where we have filters. The second major idea is that when there's an error, when we fall down, there's strong excitatory input via climbing fibers that then weakens those synapses that were active during the error. So whatever pattern your cerebellum created that made you fall over, or that made you throw a dart and miss the board, everyone laughs at you. Your olive is going to Erase those synapses. Don't do that again. Try something else. We'll see how it works. And this is why you have to practice movements many, many, many times before they become smooth and reliable. If you want to be able to shoot a free throw reliably, you don't just go try it once. You probably go practice maybe 3 times. That should be sufficient. Because the rearrangement's random. Oh, you tried this, didn't work. Oh, you tried something else, didn't work. You have to go until you find something that does work, where you don't produce the error, and then practice that again and again and again and again, so that becomes reliably activated. So let's go through our circuit, and then we'll talk about modifying it. I want you to take note of the filters along the way. This is important. This amplifies signal to noise. That's the whole purpose of having this feedback inhibition, or the lateral inhibition. If you're not strongly activated, I don't want to hear from you. That's essentially what it's saying there. We're going from analog, shades of gray for activity, to digital. You're active or you're not. So we create a clear pattern, black and white. The organization of the cerebellar cortex is in 3 layers. Unlike the cerebral cortex, which for the most part has 6 layers, except where it has 3, the cerebellum always has 3 layers. The outermost layer is what we call the molecular layer. Here we're going to find a couple of inhibitory neurons that are going to act on our Purkinje cells. Then we have our Purkinje cell layer, which is where you find the Purkinje cells. No surprise there. These are also inhibitory. What you should notice is that their dendrites are elaborate, first of all, and they are arranged in a planar fashion. So here's my Purkinje cell body. Its dendrites are organized as such. So they run within a single plane, but within that plane they are very elaborate. And this allows them to contact a large number of different parallel fibers by having that arrangement, and allows them to pack in there closely without touching each other's dendrites. So you can put in a whole bunch of Purkinje cells. If it was wild and going every which way, it'd be chaos. So to provide clearer organization, their dendrites are organized in a very planar fashion. If you go to the very first slide on this talk, you'll see the dendrites of 2 different Purkinje cells, and they rotate at 90 degrees. You can see it's just like in the cartoon. So whenever we look at this blue Purkinje cell here, notice it's elaborate dendritic arbor, and this is a simplification. The arbors are much larger than this. When we turn 90 degrees, it's flat, because they're all running this way. So they're going to encounter a large number of parallel fibers. That's all these little red dots here. This is a cross-section of all the axons created by our granule cells that live down there in the granule cell layer. So we have our outer molecular layer, then the Purkinje cell layer, and then the innermost granule cell layer. Here's our input. A bit of a filter, output. Purkinje cells are going to be the output to the deep nuclei. The granule cells are going to be the input. And the granule cells are the only excitatory neurons in the cerebellar cortex. Very easy to remember. Everything else is inhibitory. Granule cells are going to be excitatory. Also found in that granule cell layer are Golgi cells. Here's another filter. So, input, output, filter, filter, filter.
Do that again.
Yeah.
More slowly.
Input. Slower. Oh. Good. Everything else is a filter. So filter, filter, output, input, filter. And we'll see these as we go through the remainder of the slides. The input, granule cells. Here's where our mossy fibers arise. And the mossy fibers would be everything. The input from the vestibular nuclei, input from the pons, input from spinocerebellar tracts. All of those are lumped together as mossy fibers. The only thing that isn't a mossy fiber would be the input from the inferior olive. That's our climbing fibers. You'll notice that it's going to go up and climb along the dendrites of our Purkinje cells. The climbing fibers don't hit the granule cells, so they're not mossy fibers. The mossy fibers are going to terminate on the deep cerebellar nuclei, and then the granule cell as well. So there's direct input to the output nuclei here, and then we also go through our cerebellar cortex. We're going to be going through this circuit in this section here. So mossy fibers, these are excitatory. We have glutamatergic input. We're going to hit our granule cells and excite them. Granule cells make parallel fibers. These all run in parallel to one another, orthogonal to the plane of our Purkinje cell fibers. So if here's my Purkinje cell and its dendrites, my parallel fibers are all running this way. So all those Purkinje cells that are lined up in a row, your parallel fiber is going to hit all of them. So each parallel fiber is going to encounter thousands of Purkinje cells along the way. And this allows the cerebellum to create an infinite number of patterns. So you can learn anything. You can learn how to walk. You can learn how to lift heavy weight. You can learn how to throw a dart. You can learn how to juggle a chainsaw. All you have to do is practice. It's all in there. There's an infinite number of patterns to be created because of this. Because every parallel fiber is going to hit so many different Purkinje cells, we can create an infinite number of patterns. These parallel fibers, along with the mossy fibers, are glutamatergic. So here's your excitatory portion of the cerebellum. Everything else is inhibitory. These parallel fibers are going to make very few synapses on each Purkinje cell, and as such, their effect is very minimal. Very weak input to each Purkinje cell. This requires multiple granule cells to be active in order to stimulate Purkinje cells. Thus, you must have an elaborate pattern of activity. And as such, that's going to help the cerebellum then create so many different patterns. If you had a nice linear pathway where one granule cell can excite one Purkinje cell, there is no different pattern. They're both active or they're both inactive. But by having multiple granule cells contribute to each Purkinje cell and contribute to thousands of different Purkinje cells, that's going to help you create many different patterns depending on the strength of your synapses, depending on whether you had an error or not with that movement. So a bunch of weak synapses that are all going to work together to decide which Purkinje cells are active. So we're creating unique patterns here. The reason we have to have elaborate patterns is because we have very weak synapses, very few. And this is in stark contrast to what we see with climbing fibers. We'll get to those later. Now there's a little bit of a feedback here, a little bit of a filter. So down there in the granule cell layer, we have Golgi cells. These are GABAergic. They provide feedback inhibition to those granule cells that excite them. So the parallel fibers are going to hit Purkinje cells, but also Golgi cells. So they're going to start stimulating this inhibitory neuron that will inhibit them unless they are sufficiently excited to activate nearby Purkinje cells that inhibit that Golgi cell. And this is how we're creating a better signal to noise. If you're very weakly active, you're going to turn yourself off by stimulating this inhibitory neuron. So now those very light shades of gray, turn them off. Only those granule cells that are contributing to the activity of Purkinje cells remain active because they turn off their inhibitor. So when we are active enough to excite Purkinje cells and thus affect the activity of deep nuclei, in other words, if we're doing something, we're allowed to do something. And if we don't do something, then we don't do anything. It's just that simple. Low level of activity, turn that crap off. Actual robust excitation, oh yeah, leave that on by inhibiting the inhibitor. So here's a filter. If strong input, go ahead. If not, don't. Now we're going to start to create a better signal to noise. Black and white, not shades of gray. Are you active or not? I can't really tell, I don't know. Get rid of that. Make a statement. Write in ink, not in lead. Our Purkinje cells are then going to affect the deep nuclei. And they have that feedback to Golgi cells that we just went through. But they're going to target deep nuclei. These are a mixed bag. In your deep nuclei, you have GABAergic, lysergic, glutamatergic neurons. The devil's in the details. That's good news to you. You don't need to know them. You just know that they do stuff. They affect whatever they're projecting to in such a way that we can do this. We got coordinated movement. How they do that? It's a mystery. If you got more than like 3 cells involved, I can't tell you about it. Too complicated. So the input here, parallel fibers. That's the major source of input. If everything's working just fine, parallel fibers. Those climbing fibers are going to come into play when things aren't working fine. So here's a slice through our cerebellar cortex again. Here we can see our nice big blue Purkinje cell. Here's the dendritic arbor, and all those little red dots will be parallel fibers running along, making 1 or 2 weak synapses. If the Purkinje cell is excited, it then goes down and inhibits the deep nuclei. So it's going to inhibit certain neurons in the deep nuclei to make yet another pattern. So the pattern of input from the mossy fibers affects the pattern of activity in granule cells. Granule cells affect the pattern of activity in Purkinje cells. Purkinje cells affect the pattern of activity in deep nuclei, and those deep nuclei affect the pattern of activity in our motor neurons, so we do something in a coordinated fashion. That climbing fiber input is a little different. Here we're going to make a whole bunch of synapses onto 1 to 10 Purkinje cells, so a small number, but a lot of excitatory synapses. So we're going to get strong excitation here. And that's how we're going to learn. But before we learn about that, there's one more filter that we have to encounter here. So out there in the molecular layer, we have our stellate and basket cells. Basket cells are going to affect the cell body, so they'll provide inhibitory input closer to where we generate action potentials. Stellate cells are going to inhibit the dendrites. So what these are going to do is make sure that only the strongly activated Purkinje cells are activated, because they're going to provide local inhibition. So whenever we have excitatory input in this area, let's say, so parallel fibers are firing, they're releasing glutamate. They're going to stimulate our Purkinje cells, and the ones that are active are going to stand out a little more so because of the activity of our stellate and basket cells. So out there in the molecular layer, we have this little stellate cell, kind of looks like a star, and then basket cells that make a basket of synapses around the cell body. These provide widespread inhibition. So again, if you're only weakly active, you're less active, because now your inhibitors have been recruited. If you're strongly activated, you remain active. So we go from shades of gray to digital, black and white. So when you excite those Purkinje cells, their neighbors are going to be inhibited. So they stand out, better signal to noise, because you should keep in mind that most neurons are not just sitting in silence. Purkinje cells are no exception. They pacemake. They're always firing. So those that aren't stimulated by the parallel fibers, we need to turn down their level of activity. And that's what our filter is going to do. They're going to decrease the rate of pacemaking. Those that received excitatory input will have an increase in their rate of pacemaking. So rather than only providing an increase, we're providing both an increase to those that we stimulate and a decrease in activity to those that we don't. Better signal to noise. This filter here is what cuts down the noise. Those parallel fibers, when they're exciting this Purkinje cell, they're also exciting the filters next to them. So we have a burst of activity here. This neuron gets excited and all the neighbors get inhibited, because those parallel fibers stimulate our filter out here. So we get ones and zeros. We're active or we're not. And that's it for our filters. The final thing that we need to talk about would be learning. How does an error affect the cerebellar circuitry here? we have a pretty good idea about that. Now, how the cerebellar circuitry creates different patterns of motor output, well, that's pretty complicated. You can imagine how it would all work and provide some hand-wavy explanations. Here we have a much more precise mechanism for how motor learning occurs. It all has to do with whether or not your inferior olive is activated. If a movement didn't go as planned, if the input from our spinal cord doesn't match up with what we expected it to be, for example, well, that's going to stimulate the red nucleus and then the inferior olive, and then we get climbing fiber input. Here's our climbing fibers again. If we fall down, if we experience pain as a result of our motor output, we probably don't want to do that again. That ascending input from the anterolateral pathway is going to stimulate the olive, and we'll get climbing fiber input. What this climbing fiber input does is provide synchronous excitation at a large number of synapses for a Purkinje cell. Our parallel fibers will give us 1, 2 synapses, but the climbing fibers are going to give us hundreds of synapses. They're going to climb along dendrites and create multiple synapses. And when this one axon fires, all synapses are excited at the same time. That creates a complex spike. It's complex because it's not just a single action potential. The excitation was so robust that these Purkinje cells are actually excited above threshold potential. So now rest is above threshold, and you get this very sloppy looking spike here, where you get a couple of action potentials, but because you're so depolarized, you don't actually generate another action potential, but you do stimulate your voltage-gated calcium channels. So they enter essentially depolarization block there, where the excitation prevents generating an action potential, but who cares? We're not trying to communicate with our targets, we're trying to erase our synapses that created this error. So here's our resting membrane potential for a Purkinje cell. Notice it's always firing, they pacemake. Here it's turned down, it's pacemaking. So its neighbors must have been excited. That filter out there in the stellate and basket cells would inhibit it. So you get a decrease in firing rate, that's decreasing our noise. And I suppose there was an error, because here's where you get climbing fiber input. That climbing fiber input creates this complex spike. Notice the resting potential that they're measuring is pretty close to where they're spiking at. This neuron is incredibly depolarized. And if it's unable to drop back below threshold, of course you don't deactivate your voltage-gated sodium channels, no other spikes. But we're strongly depolarized. That's the main point. That strong depolarization from these many, many, many excitatory synapses all at the same time strongly excites you. Yeah.
Wouldn't you want that to continue to really make sure that those synapses are degraded? That's the problem.
Timing is everything, because you're always moving. And so you only want to erase those synapses right at that narrow window that's associated with that movement that generated the error. You don't want to unlearn everything. But you do want to unlearn whatever it is you just did that made people laugh at you or made you fall down.
So that little spike is what does that then?
Yes, this little spike. That little spike is what does it. Now, how does it do it? Well, here's how. Need I say more? So, climbing fiber input. Many, many, many excitatory synapses all active at the same time. So you have very strong depolarization. Here's your very strong, here's your very strong depolarization. We're resting far above threshold because we had so many excitatory synapses active at the same time. We're strongly depolarized, so things like voltage-gated calcium channels get open. So you get this burst of calcium. Global elevation in calcium. Calcium is an important secondary messenger, but it's not the only one. And the target of this calcium is protein kinase C. It's not protein kinase A, it's not protein kinase B, it's not protein kinase D either, or G or M. No, it's protein kinase C. Now there are two things necessary to activate protein kinase C, and one of them is calcium. How convenient. We have a global burst of calcium because of climbing fiber input. Now how do we know which synapses were active? That's the problem we need to solve here. I don't want to erase all my synapses. I just want to weaken those that made me fall down. The easiest way to know that is to ask, where was glutamate released? Which one of my parallel fibers gave me this error? I want to weaken that synapse, but not all the others. So I get calcium everywhere, but I only generate diacylglycerol where I release glutamate. And that solves the problem. And the reason that diacylglycerol is so important here is because it's a lipid. It stays right there in the membrane. It's not an ion diffusing around everywhere. It stays just right here at that synapse where we spat out glutamate and we stimulated mGlu-Rs. This would be mGlu-R1, that's a GQ coupled receptor. GQ, let me think, phospholipase C. Phospholipase C cuts PIP2 to make IP3, so we get calcium. We already had that, who cares? And we generate diacylglycerol. So when you cut that phospholipid, you get this secondary messenger here that floats around, and also this one that stays at the membrane. It tags that synapse as being active. So if you have diacylglycerol and calcium, now you stimulate protein kinase C. So you can upregulate calcium everywhere you want in the cell. You're not going to stimulate protein kinase C, though, if you don't have diacylglycerol. This is how the neuron knows there was an error, climbing fiber input. So something went wrong. Which synapse or synapses created that error? Well, the ones with diacylglycerol there. Now I have calcium and diacylglycerol, so I stimulate protein kinase C. This does what any kinase does, phosphorylates things. I have to tell you what things if I expect you to know it, so let me tell you the thing. AMPA receptors. Those ionotropic glutamate receptors that are responsible for fast synaptic transmission. When AMPA receptors are phosphorylated by protein kinase C, they are flagged for removal. That phosphorylation causes them to be endocytosed, so removed from the surface. So here's what's happened. We had an error signal, strong depolarization. We had a global increase in calcium. At those sites where we had glutamatergic input, we also have diacylglycerol. So we have the two components we need to stimulate protein kinase C. So we remove AMPA receptors. In other words, we weaken that synapse. It now has fewer AMPA receptors, so the next time we dump glutamate out, it's not going to be as excitatory. We're not going to repeat that error. What are we going to try? I don't know. It's random. We just roll the dice. Does this one work? If we don't get climbing fiber input, great. And if we do, not great. Don't do that again. Let's roll the dice again. So the working model for our cerebellum here is that some pattern of activity in the pons, in the spinal cord, in the vestibular nuclei, that's going to tell us what are we planning on doing, what's going on in our body, what's our sense of balance. So input from the mossy fibers is going to be different depending on our body position and our planned action. And those differences in input create a different pattern in our granule cells. And those create a different pattern in the Purkinje cells. And those create a different pattern in our deep nuclei. And those create some different pattern of motor output. It's all hand wavy here. There's so many different neurons involved, you can't have a precise, well, this one's going to activate, neurons 10 billion 1 and 10 billion 5. But it's different patterns. And by having different patterns here, you create different patterns here, here, and what matters to us, here. That motor output, should we do it again? Well, it depends. Did we activate the inferior olive? Did we think that we were going to have our hands clap and make a nice sound? Or, oh no, that sucks. Oh, that's much better. Yeah, I'd like to hear that at the end. We can refine our motor output based on whether there's an error. Did we fall down, for example? Did we miss? Did we not get the sound we wanted? Because if you go back, you'll see there's input from the auditory cortex to the cerebellum. It's in those maps. Slide 3, no, 4, I think. Something like that. Doesn't matter. Maybe I made an error. My inferior owl will take care of it, because what it does is strongly excite my Purkinje cells. And that strong excitation weakens those synapses that were active here. So what it does is erase this pattern. You tried that, it didn't work. Try something else. And we will continue to try something else until success. There's no reason to change it now, so don't. By practicing then that pattern over and over and over, rather than getting long-term depression, we get long-term potentiation. So we strengthen those synapses. We'll talk about that in lecture 22. But it's the opposite of long-term depression. Instead of removing AMP receptors, put some more in there. And now you have a stronger synapse. So that pattern you made here when you thought, I would like to throw a triple 20, reliably creates the pattern of Purkinje cell activity, deep cerebellar nuclei activity, and motor neuron activity, so that you throw that triple 20 time and time again. That's about it. Do we have any questions? Okay, go through these and then we'll call it a class. I really wish you would have.